Yeah, with that, I would like to conclude, and thanks again to my sponsors and Computational Medicine from UCLA. Yeah, and take any questions. Thank you. Okay.
How is the score determined? Somebody determined that it was a
Yeah, that's a standard that, so the Phred score is, the question is, how is the Phred score determined? So Phred scores are a standard that exists since, I think, 30 years already, and they're just supposed to represent how sure you are of the quality of this particular base. But you can use it, it's just as a scoring metric. It's the same as if you were to say, I am 99% sure or 98% sure, but instead of using numbers, which would take 2 spaces, you can represent those numbers as characters and have a standard scale somewhere to look up. So you know that I means that you're 99.99% sure. The next character means that you're 99.9% sure. And this scale, you can find it easily with a Google search. Yeah.
How they're actually calculated, though? Do you know?
No.
It's a mystery. Like, does somebody, how do they know that review was bad? Like, how is that?
Oh, how do they know that why are they, so so the question is, how do they know that the read is bad? The Phred score is assigned to how confident is the machine that this base that it is reporting is actually the base that it's present in the DNA. Exactly. So when the image segmentation takes place and the machine assigns one of the 4 bases to be the highest probability base there, it's also saying how confident the machine is that the base that it's calling, it's actually there.
Is it based on like the intensity of fluorescence, or?
Yeah, it's the intensity of the fluorescence, the color that it's observing, and the quality of the segmentation. Yeah, and so the farther you go on the read process, on the sequencing process, the lower that quality gets.
But overall, the exact process for how they do it is a company secret.
Which company?
Whichever company, no one actually tells you. So
Yeah.
which is why a very popular step to do in analyzing this data is base recalibration. You use like known SNPs and you try to recalibrate the quality scores using that training data. So even the companies, what they're doing, even though it's secret, it's still wrong.
More questions?
Yeah, could be. I have no idea.
What level of automation is present in like preparing the read segments and stuff? Like what do you actually feed into the computer to get to this point?
To get...
What does the machine require you to do to actually prep this? Like
Here you mean?
The library prep or the sequencing?
Sure, well like any of that. What do you
So
what part of it is done by a machine and what part like do you have to like go in there and shake it up?
right, so of all these steps, usually research labs will have people do this step, this step, this step, this step, yeah. And then here you do all of this by yourself, mostly, and then you send it to the company that is doing the sequencing, and then they will do all of these other steps inside the machine. They just put that in the flow cell, and that gets sequenced. But if you have more money and less time, then you can send your DNA to a company, and they will take care of these 2 steps, these 3 steps.
There's also robots that can do like all of it, but that's only for like really like industry labs.
Yeah, if you have everything very standardized, a very standardized pipeline, you can automate most of these steps. Yeah, all of them. Taking the sample is always done manually.
We hope.
Yeah, for now.